time getting up this morning?
Yes.
Wow. Okay. This is kind of an introductory chapter to metabolism and how organisms break down food material and gain energy. And we're going to be looking at catabolism plus anabolism equals metabolism. So the breakdown of energy sources and then the building up. So catabolic and anabolic reactions. Like I said, this chapter is a little bit of an introduction because I don't cover it in detail. You are covering glycolysis, you're covering Krebs cycle in the introductory course. You'll cover that in biochemistry if you take it. And so I'm trying to give a little bit of a big picture view, at least in this chapter, in terms of metabolism. And so, like I said, catabolic reactions are taking complicated, energy-rich compounds and breaking them down and releasing that energy, whereas anabolic reactions are taking simple compounds and requiring energy to build them up. And so, you can see that is going to be kind of the big picture of metabolism. And those catabolic reactions that release energy provide the energy that the anabolic reactions require. So you think of anabolic steroids. These are the steroids that help build muscle, requiring ATP for that synthesis reactions. So this is a modification of a slide that I've made over the years. And there's two ways that I'm kind of stressing the metabolism in this couple of chapters. The first is, this is a set of, well, they're both basically catabolic reactions. One of them linked to the anabolic. So in the first, catabolic reactions are all about stripping electrons from electron-rich compounds. And the question that has to be answered in those reactions are, where are those electrons going to? And so under aerobic conditions, they go to oxygen. Oxygen is the electron acceptor. So CH2O, this carbohydrate. So look at the word carbohydrate. The hydrate of carbon is CH2O. So that's going to be the typical base for any sugar is going to have that formula relationship. It's going to have twice as much hydrogen as carbon and oxygen. Glucose, fructose, galactose, all of those sugars fit that formula, so it's kind of convenient. It's going to have some ratio like that. And then also, hydrate of carbon tells you, there's the H2O, hydrate of carbon. So these words help us, they're meaningful. They tell us what they are. So here's your carbohydrate, energy-rich, electron-rich. So I'm really going to stress energy is associated with electrons, even more basal and more primary than ATP. So it's the electrons that go to make ATP. And so electrons are the more primal energy source. Okay, and then, like I've said, these catabolic reactions, so the complete oxidation of a sugar all the way to CO2 means as many electrons as can be stripped from that compound has been stripped, and it's completely oxidized to carbon dioxide. Generate ATP to drive these catabolic reactions. And I've given you a typical catabolic reaction where you take fatty acids and synthesize them into lipids. And so those biosynthetic reactions require ATP that are provided by the catabolic reactions. And so right here, this little framework represents basically all of metabolism. The catabolic and anabolic reactions coupled together. Okay. Also kind of big picture view of energy defined as the ability to do work. Entropy, a measure of the disorder or randomness in a system. And so that energy is required to assemble simple disordered molecules into a complex ordered form of the cell. And I say their mandate, cell's mandate, and ours is to bring order out of chaos. So here it's fairly chaotic outside the cell. There's a pretty random mixture of sugars and nitrogen sources and salts. And in the cell, it is then its duty, or this is what it does through its enzymes, its motility, its behavior, things that we've talked about in the course, to bring order out of this fairly chaotic mix of biochemicals. And so you can see there's a pretty strong disorder-order barrier right across that membrane. Semi-permeable means it is selectively taking up those compounds. It is selectively using and reducing the entropy at the cost of energy. So, kind of other way to look at metabolism, and this is kind of a little introduction to thermodynamics. Okay, and it's this kind of interesting local temporary gain of energy that enables the cell to grow, to temporarily restrain the entropy. And it requires a continual gain of energy, and then radiation of heat. Heat is this byproduct of at least cellular metabolism. And so some of that entropy escapes out as heat. Heat is a waste product, but us mammals use that waste product to maintain our body temperature. So it's not an entire waste in the case of mammals. And then there's a section in your text about, well, the whole Earth is sort of like this. It's a pretty chaotic universe, and looking at the life forms on the planet, yes, energy is dissipated as heat, but you can kind of look at the biosphere as this thermal reactor. And so it started this interesting little analogy to a cell. And some people take this to too far of a degree and start talking about, the Earth being, some sort of the entire Earth, some sort of living being itself, a single being. And okay, that takes it a little too far. But in terms of taking energy and converting it to biomass and structure, and converting it to biomass through reactions like photosynthesis and heterotrophy, bringing order out of what is fairly chaotic outside of the Earth's atmosphere. And so, I don't know, it's just a reasonable analogy. And then outside of the Earth, the energy is dissipated again, and order decreases. So we have this wonderful temporary building of order of what from outside the Earth is fairly chaotic, and then when we release energy, it becomes chaotic again. So it's just interesting how this is part of what we do, is bring order out of chaotic substrates. That's what metabolism is about. Okay, so in an orderly place, energy is conserved and entropy is reduced. Okay, So how about some of the main players is what we're looking at here. Energy carriers, and how does the cell transfer these little packets of energy, electrons. Energy carriers, the examples that we're talking about is NADH and ATP. And the key is they are able to release and transfer small levels of energy that are capturable by cells, by biology. That's one of the key factors or characteristics of these energy carriers. And NADH functions directly as an electron carrier. And so that's what we're talking about, donating electrons and accepting electrons. An electron donor is an agent that reduces other compounds. And so remember, when we're talking about electron transfer, it's a negatively charged subatomic particle that reduces the charge of whatever it receives. And so an electron donor, if you're donating electrons, you are therefore reducing the compound that accepts that electron because you're reducing the charge. So, there's some intuitive understanding there in terms of oxidation, but especially the term reduction. Reduction is the reduction of charge because it's accepted an electron. Whereas an electron acceptor is going to take electrons from other compounds, and when that happens, we use the term that compound has become oxidized. Once it has lost that electron, it is oxidized. I have become reduced because I, as the electron acceptor, have accepted this negative charge, and my overall charge has become reduced. So use those terms to help you, at that intuitive level, understand oxidation reduction, and it'll help you take away the mystery, the confusion of oxidation reduction. So here's this example, a reduced compound, and there is CH2O. There's the formula for glucose. If you know glucose has 6 carbons, you therefore know the rest of the formula for glucose. Double the 6 to 12, and you've got the same amount of oxygen as carbon. And so this is then the complete reduction of carbon dioxide, and those electrons go to go to oxygen, which becomes reduced to water. Oxygen being the electron acceptor. So those are two examples. The electron donor is glucose, the electron acceptor is oxygen. Okay. Glucose is oxidized to CO2. Oxygen accepts the electrons and is reduced to water. I think that's what I just said. But there it is in text white. Okay, adenosine triphosphate. The text gives you these structures. I'm not going to require you to copy or memorize these structures for the exam. Just want you to know a couple of features of adenosine triphosphate. For one thing, it's a nucleotide. And so to gain some appreciation of the facility that nucleotides serve in the cell, they serve as energy transfer molecules, an ATP, but as you've learned in other classes as well as this, serves as information storage in DNA and RNA. And so it's just surprising how many functions there are to nucleotides. In the physiology course that I teach, we talk about nucleotides being signaling devices, and I think I've mentioned that here in this class. So understand ATP is a nucleotide, nitrogen-containing base. You know, here is the nitrogen-containing base. Here's the ribose sugar, and here's a phosphate group. Okay, so the key to ATP is this phosphate groups that are attached to that number 5 carbon on that ribose sugar. And the more phosphate groups that are attached, and you know, we're talking about 1, 2, or 3 phosphate groups. The more phosphate groups that are attached up to 3, the more energy there is stored in those phosphate groups. And the key is to look at these negative charges that are just piling on, and there's a little squiggly red line. Because of those negative charges, it's like compressing a spring that is repulsive. So, you know, those negative charges are repulsive to each other, and when you put a second and especially a third phosphate group on that ATP, it's like more and more tightly coiling a spring, and so there's all this potential energy there, which then can be used to do work. So it stores energy in that tightly coiled spring of repulsive negative charges, and it can be used one at a time to release that energy in these discrete, controlled, relatively small levels of energy that the cell can capture, can use without, you know, like exploding. Cells don't use TNT as an energy source. Cells do use gasoline as an energy source, and we'll talk about that at the end of this chapter. Okay, like a tightly coiled spring is those phosphate groups, those negative charges that are repulsive to each other. Now, with that said, one of the interesting things that is pointed out is under physiological conditions, ATP always forms a complex with divalent cations like magnesium. And so here is your adenosine triphosphate, and they're all a little bit like bent over each other, and to some extent those negative charges are buffered or neutralized by magnesium. And so that's kind of a thermodynamic thing, that all molecules are tending towards a minimal energy source and or a minimal amount of energy, and so that magnesium allays some of that tension by neutralizing the negative charges. And those are not, you know, those are just ionic bonds that form and release, and it doesn't inhibit ATP's reactions. But, you know, that's just more for your interest, that in biological cell, magnesium to some extent neutralizes those negative charges. Okay, so looking at a couple of these electron carriers that I know you've seen before. So nicotinamide, adenine dinucleotide. The interesting thing about these electron carriers is, as I've said, they are carrying electrons, and guess where the energy is? It's in the electrons. And there's more energy associated with the 2 electrons that are carried by NAD than there is in ATP. And so there's more potential energy in those electrons, and to track energy is to track electrons, is one of the things I stress. The electrons are the basis of energy. And so NADH donates and accepts electrons, hence it's called an electron carrier. It can accept and donate electrons. So when NADH is in the reduced form, in other words, it has the negatively charged electrons, you can see that electron by looking at the hydrogen atom. And so here's a proton plus an electron. How about if we do this thought we would do this with 2, becomes hydrogen gas. And my real point is really a single electron to show it to you. When a proton, which lacks an electron, accepts an electron, it's a hydrogen atom, and sometimes that's abbreviated like this. In other words, there is the electron. And what I'm trying to point out is to know where those electrons are and to know whether this NAD or NADH is reduced or not, whether it has the electron or not, is simply to track the hydrogen. The hydrogen tells you that this electron carrier has accepted an electron and is reduced. It is now carrying that electron because basically in a biological cell, electrons are transported with a hydrogen atom. So anyway, I don't know if all that was worth writing out. Here is NAD plus, 2 protons plus 2 electrons. One slightly interesting thing about NADH is it only accepts 2 electrons, but only one of the hydrogens. So it's NADH without any kind of a positive sign. That positive is on the NAD plus. Okay, so overall reduction of NAD plus consumes 2 hydrogen atoms to make NADH, but it only really carries one of those hydrogen atoms. And so this is what I keep saying, catabolism is where electrons are stripped from glucose and now are being carried by NADH. They don't go directly to oxygen. There's this intermediary called NADH. Okay, so here's the NAD structure. And the only point here, you don't need to know these structures, is to see on this nitrogen-containing base that this is where those electrons are accepted. So it's on this ring that is aromatic at this point. So there's alternating double bonds. When they accept 2 electrons, that ring has now been partially saturated, and so it's no longer completely aromatic. There's not 3 rings in this ring anymore because it's accepted 2 electrons. And so there's the presence of that 1 hydrogen atom that you now call this molecule NADH. So that's a little bit of details. Certainly the structures for your information. Probably so is that detail. Okay, FADH2, you've seen this as well, is another electron carrier. Flavine, adenine dinucleotide. So we call these coenzymes because they assist enzymes to carry out reactions. So FADH2 is the reduced form, and FAD is the oxidized form. So a little different than NADH. It actually, you can see both of those hydrogen atoms on the ring, and there they are. Again, kind of reducing the aromaticity of these rings of the flavin. But you'll get that in a biochemistry course. Okay, you've seen this chart I don't know how many times, but it's helpful just to remind you of what enzymes do. This is basically the amount of free energy in a system that anytime you carry out a reaction, there's this hump that you have to overcome to then release the energy. And so you go from a state of high energy to low energy. There's this delta G, which is the release of energy, and the bigger the delta G, the bigger the amount of energy released. Enzymes simply reduce this activation energy. So here's the energy required with an enzyme, which is smaller than the energy required without an enzyme. And so this activation energy prevents so many reactions from happening spontaneously. Glucose in water doesn't spontaneously get oxidized to pyruvate. It requires energy. It requires enzymes to catalyze those reactions. And so that's a good thing. Things are not happening spontaneously. Part of the order that has been brought to the universe, to the cell, to the earth. You can take that paradigm and blow it to the ends of the universe and bring it to the subatomic particles. It's really, it's great. Okay, so here is a pretty nice diagram in your text that is shows you a typical enzyme involved in catabolism of glucose, and it's pyruvate kinase. And here are these active sites of the two substrates. So phosphoenolpyruvate and ADP is the active sites, the sites that are binding the two substrates are at one part of the enzyme. But this is a special enzyme, and many enzymes are special like this, and that is many enzymes are allosteric. So they have a binding site for some coenzyme that is involved in the reaction, helps stimulate this reaction. So the coenzyme here, or the cofactor, is probably the better way to describe this, is fructose 1,6 bisphosphate, this allosteric site. When it binds to that allosteric site, it changes the conformation of the enzyme to then let this reaction go to completion. So here's this sort of barrier between the two substrates without binding of the fructose 1,6 phosphate. It binds, that barrier is no longer there, and the reaction proceeds to completion. And so phosphoenolpyruvate and ADP now immediately go to pyruvate and ATP. And so typical nice illustration of an allosteric enzyme, and here's the real model of it in terms of showing you what those sites really look like. Okay, kind of big picture view of catabolic reactions. I'm always putting this C6H12O6 going to C to 6 carbon dioxides. 24 electrons are harbored in glucose that are sequentially released through the reactions of glycolysis and tricarboxylic acid cycle, the TCA cycle. And it's the capturing of these electrons, which is all about then the conversion of those electrons into ATP. So this is just to give you an appreciation for, well, all the things we eat are all the things that bacteria eat. So here's sources of lipids, here's sources of protein, here's sources of carbohydrate. Kind of the interesting thing in this bottom shelf of substrates are some of the unique substrates that are used by bacteria. So they can use these very complex and at times toxic compounds. We've got organisms in my laboratory that break down trinitrotoluene, TNT. There's organisms in nature that break down these massively complicated lignin molecules that would otherwise never be broken down if it wasn't for these lignitic fungi, these white rot fungi that live in the woods. So just a little bit of an expansion of not just the usual culprits are used as substrates for energy. Okay, this is a big picture view that I'm really only stressing a couple components of it, but at least see this big picture, and that is all these different substrates are funneled into, say, single pathways, like glycolysis. And they enter glycolysis and the TCA cycle at different components. So the sugars go directly into glycolysis, but lipids and glycerol enter it at different points. So fatty acids come in at the beginning of the TCA cycle. And so all of those different components that we just looked at in the last slide are funneled into kind of the usual culprit pathways that are utilized. And so just to point them out, glycolysis, glucose is catabolized through glycolysis, step number 1, through the TCA cycle, step number 2, and then the subject of the next chapter is respiration. And so I just point out 2 of the 3 main cycles that are used to break down almost any compound under aerobic conditions, glycolysis and TCA cycle on this chart. Besides that, and just the appreciation for these compounds entering at different points, there really isn't any information that I'm specifically after. Just a big overview figure, and understand the 3 steps: glycolysis, TCA cycle, and respiration that we'll be kind of seeing. Okay, one of the things that was added in this 3rd edition that I thought was helpful because it touches on something that we've talked about before, and that is bacteria are so small, 1 micrometer cells, and some of the things they eat are 100 times bigger than they are. So here is a photomicrograph of Bacteroides, is the name of the genus of this organism, in our gut that are colonizing these massive sources of carbohydrate, huge polymers, and they're 10 times bigger than their cells. So we've talked about enzymes that are on the outside of the cell that break down these big molecules. So this is just a nice example of that in this starch utilization system, SUS, has this series of enzymes. So Bacteroides, this genus Bacteroides, is a gram-negative bacteria, and so it has this outer membrane and then this periplasmic space. On the outer membrane are a series of enzymes that are connected with, do I write this in? A series of enzymes that are connected with an active transport porter that the enzymes break down this big polymer, and starch is a good example of these massive polymers that are bigger than a bacterial cell, break them down into smaller polymers. Okay, so now you've got a series of 4 ringed carbohydrates, and this is now enough to be taken up by this transport protein into the periplasm. In the periplasm, there's a couple of other starch utilization enzymes, these amylase enzymes, which now break down these 4 mers and 6 mers down into 1 and 2 molecule carbohydrates, monosaccharides and disaccharides. And now we've got a monosaccharide uptake enzyme, and now you've got your sugar molecule in the cell. So here is the beginning of metabolism. Here's the beginning of glycolysis, is you've got to tame what's out there to then give it your substrate for glycolysis right here. So glycolysis starts here, but understand the real world is a lot more complicated than just a lot of sugar outside the cell. It's typically it typically looks like this. So starch utilization system is a great start. Is the beginning of metabolism. Is before glycolysis. Is one of the points I'm making. Okay, this is, I'm not covering very much of this figure. Okay, so really what I want to cover is that bacteria have 3 different routes of using carbohydrates. Okay, 1 is glycolysis. 2 is the Entner-Duderoff system, which is happening in our GI tract. Okay, so if we're interested in our metabolism, well, we are interested in the Entner-Duderoff pathway because that is what is commonly used by the gut microflora in our GI tract. And then the third one is this pentose phosphate pathway, which is involved in biosynthesis. So, in kind of diminishing amounts of information, I want you to know, especially glycolysis. And what does that word mean? It means the lysis of glucose, glycolysis. So that 6-carbon molecule, 6-carbon glucose, is being split into 2 3-carbon molecules of pyruvate. And so glucose is the substrate of glycolysis. 2 molecules of pyruvate is the product. And I'm not even talking about the various intermediates of glycolysis. I just want you to know what glycolysis is. Glycolysis is the splitting of that 6-carbon molecule into 2 3-carbon molecules. Okay? Edner-Deuteroff, just know that's what our GI tract microorganisms use instead of glycolysis. They use glycolysis also, but Edner-Deuteroff pathway, which, okay, has some differences. Basically, instead of glucose, it uses sugar acids, but funnel in to end up with 2-pyruvate. You end up with the same product of glycolysis. And then just simply the pentose phosphate pathway is mostly used in anabolic reactions. And so in biosynthetic reactions. And so that's just basically introducing to you at a kind of a cursory level 2 other pathways that catabolize carbohydrates, the Edner-Deuteroff pathway and the pentose phosphate pathway. And then with a little bit more detail with glycolysis. Okay, this also big picture. You wouldn't believe how many figures I'm skipping in this chapter. We're not looking at any of the intermediates and enzymes of glycolysis or TCA cycle. But I want you to put things together, and this is a nice figure that puts them together. Okay, so kind of our model reaction, the splitting, the lysis of glucose, glycolysis is right here. Ultimately, it goes to 6 molecules of CO2. Here is glycolysis, and that is substrate product, glucose, and 2 moles of pyruvate. At the level, I want you to track how many ATP, is one of the points of this figure, is produced in these different steps. So we are looking at 3 steps in metabolism once you get the molecule inside the cell: glycolysis, TCA cycle, and respiration. How much ATP is produced at each of those levels? There's only 2 ATP produced in glycolysis. There is only 2 ATP produced in the TCA cycle. And then in the 3rd step, respiration, gosh, there's 34 moles of ATP produced. So that's why there's a whole chapter devoted to respiration, because that's where the bulk of ATP production occurs through this electron transport system in the membrane. So that's where the energy is. So let's take a quick look. There are, of those 24 electrons that are in glucose, 4 of them are carried by 2 molecules of 2-NADH from glycolysis. 2 other moles of NADH are carrying 4 electrons in this kind of transitionary step between glycolysis and the TCA cycle. Most of those electrons now are finally stripped out of these 2 cycles of the TCA cycle to completely oxidize CO2. There is your product of CO2. So here, 6 moles and 2 moles, 6 moles of NADH and 2 of FADH2. Those are now 16 of the 24 electrons are being carried. So most of the energy still is in the form of electrons at this point. You've only produced 4 ATP. So then how do you make a whole bunch more ATP? It's all these arrows going to the electron transport chain, the ETS, the electron transport system. and respiration, oxidative phosphorylation, where ATP synthase is used to make all those ATP. We'll talk about that in some decent detail in chapter 14. But, hey, there's the big integrated picture of glycolysis, TCA cycle, and respiration, producing the two things we're interested in, ATP and NADH. Those electron transport carriers are holding the bulk of the energy. Most of the energy is in the electrons. Okay, so kind of big picture is really what I'm emphasizing in this chapter. And then we're going to finish up in the next couple minutes on some of the unusual compounds that bacteria can break down. So I mentioned the white dot fungus in the woods degrading the huge log. They're recycling the lignin. We're thankful for them, otherwise the logs would stay there forever and just get like petrified. Toxic pollutants are degraded. This is the subject of about a third or even a half of a class that I teach called Applied and Environmental. Microbiology is, biodegradation. And so we're going to look at some gasoline degradation. I said benzene is used as an energy source. Gasoline is used as an energy source. When they do that, catechol is an important intermediate between these gasoline compounds and kind of more cell-friendly compounds that can then enter the TCA cycle. So, here are the active ingredients in gasoline. So, there's a lot of toluene. I don't know why benzene is not shown here. Benzene, toluene, I guess this is a little bit more generalized. There's a lot of oil-based pollutants in the environment, and many of them are cycled to this central intermediary called catechol. And then the key step in breaking down these energy-rich aromatic compounds is to break that aromatic ring. And so the breakage of this ring right here, it has now been opened up, is the key step, and it's carried out by this catechol dioxygenase. And so once that ring is broken, then it can go down to, like I said, kind of more cell-friendly compounds like pyruvate, enter TCA cycle, just use our known pathway. So it basically, bacteria have a number of additional enzymes that we'll never invent in our body that can use toxic compounds, these various dioxygenase enzymes, bringing them to a catechol, break the ring, and once that ring is broken, okay, enter it into the TCA cycle, and you're off scot-free. You're biodegrading pollutants. That is what the bacteria are growing on. They're energy rich. So oil is energy rich. All of these compounds are energy rich, and bacteria have the ability to tap into that energy source. I would say, that is the main structure that, and I'm not even going to ask you for that structure. Just understand, once you break open that aromatic ring, you basically find yourself quickly into the TCA cycle. Okay, I guess I will cover this at the very end, at the very beginning of next period. I still have some exams that you can pick up, exam number 2. So if you haven't picked up your exam, come and find it in this small file. See you Wednesday.